Good morning, Biochemistry Simplified family. Madam Winnie here. I would like to discuss with the Bpharm students the cut to which they did for population genetics as they prepare for exams this week. I have to discuss how to answer the questions so that they can avoid the mistakes, the small mistakes which they are making. In any case, they find the questions or a sample of the questions which relate to whatever they did in the cut, then they can avoid, or it can prevent such mistakes to be made. So to my left is the question paper which the students have, and to my right is the marking scheme. So I'm going through all the questions, and you'll be able to see in the marking scheme keenly how you are supposed to answer the questions. So it might not be clear on the screen, but as you go read on your own, you'll see the question, or you'll have the question paper and relate to the answer sheet or the mark sheet, the marking scheme. You're going to see I've just numbered them based on what you're supposed to answer. So question 1 said, this is basically for the genetics, the population genetics. Bpharm students who did the cut a week ago. Question number 1 says, a population of 100 individuals is tested for their genotype at a particular locus R. Observed frequency were as follows: homozygous dominant 55, heterozygous 20, homozygous recessive 25. Determine the allele frequency and use a chi-square test to determine whether the population is in Hardy-Weinberg equilibrium. Using a degree freedom of 1, remember we have 2 alleles, biallelic. So degree freedom of 1 at a value of 3.84. You have to prove that the population is in Hardy-Weinberg equilibrium. And I mentioned that in Hardy-Weinberg equilibrium, the genotypic frequencies should add to 1. The allele frequencies should also add to 1. So at that point, we are talking about the population is in equilibrium. But at the point where the genotypic frequencies are not adding to 1, neither when you calculate all the genotypic frequency, the chi-square value is greater than 3.84, then you'll be able to say that the population is not in equilibrium. Thus, we are going to reject the null hypothesis. So at that point, let's go straight and see what you're supposed to do. In fact, most students did well by showing me how to calculate the genotypic frequencies. If you look at my marking scheme up there, I've mentioned question number 1. What you do for the genotypic frequencies, get a 55 over 100. For the homozygous dominant, you'll get 0.55. Heterozygous 20 over 100, you'll get 0.20. You can see how you are getting the marks. For the homozygous recessive 25 over 100, you get 0.25. Some students were not showing me the genotypic frequencies. They go straight to allele frequencies. So why the hurry? Because you score less marks. So you have to show the genotypic frequencies, because I told you for you to get the allele frequencies, we have to start from the genotypic frequencies so that it can allow us to get to the allele frequencies. In mathematics or in sciences, show systematically how you got the value, and that value led to you finding the next value. So then that one made us to get the allele frequencies. Remember, P will always represent the dominant allele as Q represent the recessive allele. So for P, we'll get 0.65 because 0.55 purely homozygous dominant, but the traces of homozygous dominant are in heterozygous, so you do half of the heterozygous. Half of 0.20 is 0.10, and if you add it to 0.55, you'll get a 0.65. You can go ahead and do 1 minus 0.65, then you get 0.35, or you still do Q, which represent the recessive allele separately, by doing 0.25 and half of the heterozygous, because the traces of the recessive allele are in heterozygous. So you do 0.25 plus 0.10, you will get 0.35. So you'll get the allele frequencies of 0.65 for P and 0.35 for Q. Remember, you've been told to find the chi-square fitness test. Is our population is in equilibrium? And remember, the formula for chi-square fitness test is observed. Yeah, that is genotypic frequencies minus expected. Genotypic frequencies over expected. But remember, the observed and expected should be squared. So makes the formula to be observed minus expected squared over expected. For you to calculate the expected, that is where you need the allele frequencies. So what you do with the p squared, just do 0.65 squared times the total number of people in the population, 100. 2pq the same, 2 times p, 0.65 times 0.35 times 100. Q squared, 0.35 squared times 100. You'll get expected genotypic frequencies. P squared, that is homozygous dominant, will be 42.25, 2pq will be 45.50, and Q squared will be 12.25. Then substitute. Observed is what was given there. Some students were making mistake by now giving me 0.4225 because they skipped multiplying by the total number of people in the population, and that will give you a wrong answer. So if you do observed at 55 for the homozygous dominant minus 42.25, the expected, which you just calculated, squared over the expected 42.25, plus heterozygous observed is 20 minus 45.5 expected squared over 45.5. Lastly, homozygous recessive observed 25 minus expected 12.25 squared over 12.25. Remember, the genotypic frequencies for the observed were adding to 100, the total number of people in the population. Same with the expected, they should add to 100, they can't be less. We are talking about the genotypic and allele frequencies remaining constant in the coming generations, how the Hardy-Weinberg talk about it. So after finding the expected and substituting the values, you are going to get homozygous dominant 3.85, already excess because the ratio should be 1 to 2 to 1. Heterozygous already excess 14.29. Homozygous recessive very excess 13.27. Then the chi-squared value was at 31.41, meaning it is greater, far much greater than 3.84. So it's always should be less than 3.84. Then we'll always accept the null hypothesis that population is in equilibrium. But now it is greater than 3.84, reject the null hypothesis. The population is not in Hardy-Weinberg equilibrium. Question number 2. Well, only 2 students got of the 80 something, because sometimes you have to be given a value which when you calculate, you just get that the other 2 are already 1. The other two alleles are already 1. So it means the other one cannot be more than 1, but it has to be 0. So the question says, a multi-allelic system of blood group ABO, 1,200 individuals sequenced, 500 were BB, 200 were OO. Calculate the genotypic and allele frequencies. Remember to put a Punnett square so that you can express the multi-allelic system. So PQR, those are the alleles which are representing the ABO blood group system, and we'll always give A to P, Q, we give it the B, and R, blood group O. And you're told that 500 were BB, meaning Q squared is 500, R squared is 200. But that is not what we need. We need the allele frequencies. So you have to find the square root. Put 500 over 1,200 and find the square root. Then you'll have Q. 200 over 1,200. Get the square root. You'll get R. But already, if you do the 2, you already get Q as 0.64. R as 0.41. Already that is 1.15. It is not at 2 because they should add to 1. Sometimes we just bring different values to see if the students get the concept. So if you have the multi-allelic system, remember those alleles with the values you're given, the frequencies, it means they are squared. So we need the individual alleles. So get the square root. And that one was making P to be 0. So some students were already thinking of just getting the division, the answer after dividing, and then they try to put it to 1. That one doesn't work. You have to get the square root of the individual alleles, which are, the values have been given. So if you get Q and R at 0.64, 0.41, remember the allele frequencies, when there are 3 of them, they should add to 1. So it means our P will be 0. And that one will bring the P squared to be 0, because any number times 0 is 0. Q squared will be 0.41. R squared will be 0.17. 2PQ, any number where the P is times 0 is 0, it will be 0. 2RQ will be 0.52. 2PR will be 0. As much as it brings our total value to 1.1, not bad. But remember to get the square root of the 2 alleles so that you can find the third allele. It is in Q squared, R squared. But we need R, we need Q. So we need to get the square root. After getting the square root, minus, total the 2 alleles, then minus by 1. If you minus, you'll get the other allele which you don't know. Question number 3. And that is where the students are making a lot of mistakes. And kindly follow this revision, because I want us to avoid any mistake which can come during exam. So it says, in humans, the Rh factor genetic information is inherited from our parents, but it is inherited independently of the ABO blood type alleles. Independently. The law of Mendel of independent assortments. They talk much about independently. The 2 alleles are inherited independently. In humans, the Rh plus individuals, they have the Rh antigen. Remember the H small on their blood cells, while the Rh minus individuals do not have. That is the group we are interested with. The ones who do not, the ones who cannot taste, the ones who have a problem. That is what we are calculating. We are not calculating the ones who are fine. So there are 2 different alleles for the Rh factor, known as Rh plus and the small r, small h. Assume that the dominant gene Rh produces the Rh plus phenotype. Those are the people who are capable, We are not interested in the people who are capable. We are interested in the people who are having difficulties, the people who they have a problem. So we are not interested in the Rh plus phenotype. Let's proceed reading. And the recessive R small, H small, allele produce Rh minus. We are interested in Rh minus phenotype. So they are saying in a population that is in Hardy-Weinberg equilibrium, if 120 out of 500 individuals are Rh minus, that is what I want. The students were going as far as doing a square root to 380 over 500. Nobody wants that. We only concentrate on the group which has a problem. So kindly, if you see such a question, we are trying to calculate the heterozygous. That recessive allele, which is in heterozygous state, it is still in the heterozygous as a genotype. The recessive allele, which is causing such problem of Rh minus, is what we are looking for. So the 120 out of 500, get 120 over 500 and do the square root. Then you are told to calculate the frequencies of both alleles. Very simple. Calculate P and Q. And if you do your square root, if you divide it, you are going to get 0.24. If you divide 120 over 500, you get 0.24. If you do the square root, the Q will be 0.49. But then, the law of Hardy-Weinberg says that the allele frequencies should total to 1. So you get 1 minus the Q for you to get P. 1 minus 0.49 was giving us 0.51, and P will be 0.51. So the allele frequencies Q is 0.49 and P is 0.51. Question number 4. There are 100 students in a class. 96 did well in the course. The 4% blew it totally. 4 out of the 100 blew it totally and received a grade F. 96 of the 100 students are fine, no problem. But we have to concentrate on the 4 who failed. We are not concentrating on the people who passed. Why do you have to find the square root for 96%? No. We need the square root for the 4 who failed. 4 over 100. Find the square root. We are trying to look at that recessive allele in heterozygous. How many of these generations in this population will have the recessive allele, which in most cases or in some cases might be inherited? Then we can find how many of those individuals can have that allele for not understanding well until they fail. So it still says, sorry, in the highly unlikely event that these traits are genetic rather than environmental, because remember it's GE, gene or environmental, it could be the conditions where they are exposed to learn is not conducive, which made them fail. That could also be a contributing factor. It could be the students were going through a lot, but they had nobody to talk to. That made them fail, so you can't judge. But they're saying, in case where it could be heritable, that is, it's not environmental, if these traits involve dominant and recessive alleles, and if the 4% represent the frequency of the homozygous recessive condition, calculate the frequency of the recessive and the dominant allele and the number of heterozygous individuals in the population. Take 4 over 100 because it's percent. Find the square root. We are trying to find the square root of only the 4%. Then you will have the square root of 4% will be the square root of 0.04. You will get 0.2. 0.2 will be Q. If you get 0.2 minus 1, 1 minus Q will give you P. So P is 0.8. You are told that you get the individual allele frequencies, which are now Q is 0.2, P is 0.8, and then the question also asks and says the dominant and recessive allele, we've got that. The number of heterozygous individuals. Why do you have to put percent? Human beings are not percent. Individuals, you have to count 1, 2. Tell me these are the number of individuals who are heterozygous. Some students were leaving it at 32%, but this was done well by most students. So whatever you get, just multiply by 100 because the students are 100. So you are going to do 2pq, 2 times p is 0.8 times q, which is 0.2. You will get 0.32 times 100 individuals or 100 students. You will get 32 of the students are heterozygous. They have the traces of allele being the one for the failing. Question number 5. The ability to test PTC is due to single dominant T. To test, if they can test PTC, we don't have any problem. They don't have the problem of their testing bags missing something for them to test. If the test bags already can test, we don't have any problem. If most of them could test, then we don't have problem with the people who test. So 138 could test, could detect the beta test. Why are you still dividing the 138 by 215? We don't need to divide the 138 by 215. The 138 are fine. We need only to divide 77 over 215. And it is q squared. Find the square root, kindly. The students were dividing 138 over 215. 77 over... If you are confused already, then where is the answer? I was not marking any. If you do the square root, 138 over 215, 77. No, you have to decide. You have to understand the equation well and know what we are answering. We have to calculate the heterozygous. Remember, we are looking at the recessive allele, which makes people not to test. So do the q squared, get 77 over 215 and find the square root. And if I did that in my marking scheme, you can see it very well. We were talking about 77 over 215 square root, you'll get 0.598. P will be 0.402 because you'll minus and you'll be able to get P. After that, I said calculate the expected genotypic frequencies. Is that what the question says? Calculate allele frequency of each allele and expected genotypic frequencies. The students didn't get that one right, though a few got it right. After getting Q and P, expected is very simple. Just get P squared times 215, Q squared plus times 215, 2PQ times 215. It was bringing expected. P squared we were getting 34.74, Q squared we were getting 76.88, and 2PQ was 103.37. Remember, they should add to 215, just as observed, could add to 215. So that was the answer. Simple. Get the allele frequencies. After you found the Q, do the Q squared. The people who are having problem, the difficulty, the ones who are fine is okay, but the ones who have a problem, even in the case where you are asked 1 out of 500 kids are born with phenylketonuria, do 1 out of the 5,000 square root. You're not looking for the 4,900 and something. No. The ones where there is a problem. We are trying to find the traces of this recessive allele which resides in the heterozygous. Question number 6. Selection model where the homozygous dominant is selectively disadvantaged. So we talk about natural selection, which should not happen in the Hardy-Weinberg equilibrium. Is one of the evolutionary causes which cause a change in the allele and the genotypic frequencies, as we looked much into those assumptions. But at the point we are looking at the selection models, we talked about the fitness value. A genotype which can represent an individual which can represent its genotype in the next generation is given a fitness value of 1. The one which cannot represent its genotype in the next generation is given a fitness value of 0. We looked at several selection models and types, which I'm not going to discuss at this point, but I'm going to go straight to the question. It says, a selection model where the homozygous dominant are selectively disadvantaged. It means the selection will always remove the homozygous dominant, and the ones who will be favored are heterozygous, homozygous recessive, which fits well the CCR5 gene, mutant gene, the 32 base pair mutant gene, which was much developed in the Europe back then in 1900s. But then we saw that the individuals who have both genes, that is, homozygous recessive, will be resistant to HIV infection. They have immunity for HIV. The ones who will have traces, that is, the heterozygous, will be partially immune. But the homozygous dominant do not have traces of the CCR5 allele, so they will die whenever they are exposed to the virus. Some students were giving me sickle cell. Sickle cell doesn't fit in this model. Sickle cell fits in a model where both homozygous dominant and homozygous recessive are selectively disadvantaged. They are removed from the population. Only the heterozygous survive. So you are to give a CCR5. The CCR5 gene model. So based on how you describe it, don't write a lot of stories. I told you not to write a lot of stories. Just explain how the model works. Which one has a fitness value of 0? Which one has a fitness value of 1? And then give me an example, any of the diseases which relate to the model, and explain well how the homozygous dominant is eliminated, the homozygous recessive is retained in the population together with the heterozygous, if it's that model. So we have to understand the question well. The last one was inbreeding. And the mistake which the students were making in inbreeding is they will always put the formula for the chi-squared. We are not going that far for the inbreeding. You are just focusing on the heterozygous only. So we need to calculate the expected heterozygous only. So we are saying in a population of fish, the color red dominant to white, sample of 375 fish is observed, and the following frequencies are noted: homozygous dominant 39, heterozygous 154, homozygous recessive 182. Determine whether the population is inbred or not. Not in Hardy-Weinberg equilibrium, using the chi-squared, degree of freedom, no. The population is inbred or not. Simple. Start by getting the genotypic frequencies. We go straight away. Put 39 over 375. You'll get 0.104, 154 over 375 for heterozygous. You'll get 0.41. Homozygous recessive, you do 182 over 375. You'll get 0.485. That's already earning some marks. Then get the allele frequencies. Remember, the whole of 104, 0.104, plus half of the heterozygous, where you'll get 0.206 plus 0.104, you will get P or R as 0.31 and Q as 0.69. So the allele frequencies for P was 0.31, Q 0.69. Get the expected heterozygous. Not genotypic frequencies, get the expected heterozygous. You'll do 2PQ times 375. You've had your P as 0.206, Q, P as 0.31 and Q as 0.69. So if you do 2PQ, you'll do 2 times 0.31 times 0.69 times 375. You are going to get 160.43. When you get 160.43, that is expected heterozygous, go to the formula. The formula of inbreeding. 1 minus observed heterozygous over expected. If you do observed over expected, you'll get 154 minus 1. Divide by 160, sorry. You'll get 1 minus 0.963. So the value after subtracting from 0.963, you'll get 0.037. Remember, for the inbred population, I said, if the value is greater than 0.5, then the population is inbred. But it is less than 0.5. It is 0.037. So you conclude that the population is not inbred. Reject the null hypothesis that the population is inbred. So, students, kindly revise. Revise these questions, follow this video, do revisions before the date of exams, and it will be helpful. Thank you so much, and all the best with your exam.